So thanks for joining us today. I'm Rex Malmstrom. I'm a scientist at the JGI, and I'll be hosting today's session where we plan to highlight some of the capabilities of the JGI and explain how they can be accessed in order to advance your own research. It's our hope that this presentation will provide an opportunity to get answers to your questions, whether they're specific about the technologies that we're talking about, or perhaps more general questions about how to become a user. Questions will be collected today through the chat bar, and we'll take some time at the end of each presentation to address those questions. And we'll also capture all your questions and make the answers available online after the fact if we didn't get a chance to answer them in person today. And so they'll be there for you to look at later and for anyone else who cares to look at what happened. But before we get into that, I just wanted to tell you a little bit about the Joint Genome Institute. Great, you advanced the slide already, great job. The Joint Genome Institute is a U.S. Department of Energy user facility, and we're located in Lawrence Berkeley National Lab. We're a world leader in the field of genomics of plants, fungi, algae, and complex environmental microbiomes. And we help researchers characterize how these organisms and how these communities carry out complex biogeochemical processes, as well as how these organisms can be harnessed for sustainable biofuels and bioproducts. So again, we're focused on projects related to things like biogeochemistry, biofuels, bioproducts. So studies that focus primarily on biomedicine or agricultural food crops probably wouldn't be appropriate for the call for proposals, the specific call for proposals we're going to talk about more today. Could you please advance to the next slide? Great. So the JGI has resources and expertise in the areas of DNA and RNA sequencing, DNA synthesis, metabolomics. We have data analytic capabilities. And these capabilities are all provided to researchers for free based on a competitive peer review process that we call our Community Science Program, or CSP. And this is important because this is a little different from other calls for proposals. So in contrast to other agencies where an accepted proposal is awarded funding, when it comes to the JGI, a successful proposal is awarded free access to these resources and expertise that are used to help carry out the proposed work. So it becomes it sort of establishes a collaboration with the JGI when a proposal is accepted. Okay, next slide, please, Dave. So today, we'd like to share with you some information about an upcoming request for proposals in the area of functional genomics. This is an opportunity, this opportunity is focused on projects that leverage the JGI's capability to understand the functions of genes and genomes. You can see on the slide, there are a number of different capabilities available in this call for proposal, but today we're just gonna focus on two of them. Can you advance the slide? There we go. We're focusing on the synthesis of genes and pathways for functional characterization, as well as the synthesis of guide RNAs. We're going to hear from JGI scientists, Yasuo, Yoshikuni, and Blaby, and they're going to describe a little bit about these technologies. And then we'll also hear from two current members of the JGI user community, Alper from the University of Texas, Austin, and Philipp Zerbe from the University of California, Davis. And they're going to share with you how they've been utilizing the JGI's capabilities in order to advance their own research goals. On this slide, I'd like to point out that the submission deadline for this functional genomic call is approaching quickly. It's July 30th. But we also do this particular call twice a year. And these capabilities are also offered through the annual CSP and the FICUS calls, which come in the springtime. So if you want to learn more, check out the link at the bottom of the page for the current call. And then feel free to reach out to Blaby, his contact info is there, or Yasuo Yoshikuni, or any of the panelists if you have any additional questions. Okay, again, as we go, I encourage you to submit your questions via the chat bar, and we'll do our best to address them today at the end of the presentations. And these questions can be for our guest speakers as well as the JGI staff. They can be about science or about the process. Okay, next slide. So that is it for me. We're going to hand it over now to Blaby, and he will tell you a little bit more about the first technology.
Okay, thank you, Rex. Okay, so hopefully everybody can see my slides. Good morning, everybody. My name is Ian Blaby. I'm the DNA synthesis lead at JGI. I just wanted to start by describing our mission, which is basically to help our collaborators and our users to harness the power of DNA synthesis, strain engineering, and biosystems design towards DOE mission. So we have 4 scientific strategic focus areas in support of this mission, as indicated here on the slide, and really just for time reasons, we're just going to focus firstly on high throughput functional genomics this morning, and then secondly with genomes to structure to function. But I just want to point out that all are equally applicable to the core that Rex just mentioned. So to give a little background, broadly speaking, we have 4 product types that are currently supported by the DNA synthesis platform. And I'm just going to run through these very quickly. So firstly, we offer to users a single or a few small proteins inserted into constructs, and these are typically used by users for protein characterization or structural characterization. And Yasuo and Philipp Zerbe will be talking about that momentarily. Secondly, we offer the opportunity to put pathways or operons into constructs, and this is typically used for metabolic engineering projects by users. Thirdly, we offer combinatorial libraries or Golden Gate libraries, and this is typically associated with pathway optimization or screening. And then finally, we offer to users libraries with high degrees of variance. And these would be libraries that would contain tens of thousands or hundreds of thousands of different inserts. So this could be used, for example, for promoter screening. But recently, over the last year or so, we've seen a rising popularity amongst users for taking advantage of this product type for guide RNA libraries, basically enabling the interrogation on a genome scale of gene function. And so in recognition of this popularity, the JGI has constructed an end-to-end pipeline to work with our users in the interest of interrogating gene function on a massive scale. So we have our custom in-house software enabling us to work with our users in order to design guide RNAs. This could be done in relatively low complexity libraries if users want to target specific processes or groups of genes or gene families. Or this can be done on a massive scale, on a genome scale. And our software also enables us to offer redundancy within with multiple targets per gene, so that we ensure that we can capture the full genome if that's the research question. We'll then work with the user to insert the library of guide RNAs into their CRISPR technology or machinery of choice, whether this is CRISPR-A or CRISPR-I for knockdown or activation of genes. We will QC that library once we've completed the cloning to ensure that there is no significant skew in the population of library with any sort of bias towards particular guide RNAs. We then provide this as a library back to the user so that they can then take this library and transform it to their organism of choice and perform whatever screen that would address their research question. Once they've gone through several cycles or several generations, thus enriching particular knockdowns or knockups, they will send the cell pellets back to us, and then we will sequence that library in order to analyze for enrichment or loss of any particular genes, strains within that population. And basically what this enables us to do in collaboration with the user is be able to pin, depending on the condition imposed upon the culture, be able to assign or some sort of a census to the function of genes on a massive, potentially genome-wide scale. So again, just to reiterate this is very much a to and fro collaboration with users for this product type, in that we work with them to define targets and to design the guide RNAs. We'll construct a library, we'll perform the library QC. The user then takes this library and performs their screen on it, and then sends us back the pellets so that we can work with them on the sequencing. And we have the analytical tools in order to aid with the enrichment and the counts associated with that. Oops. So with that, it is my pleasure to be able to hand off to Hal Alper, who is a professor at the University of Texas at Austin. Hal has been a long-term user of JGI for many years, but in particular with relation to this particular product type, he has had several CSPs with the purpose of performing functional genomics in yeast and fungi. And with that, I'll hand off to Hal. So please take it away, Hal.
All right, well, thank you very much for that introduction. So with the short time that I have today, I really want to be able to showcase a little bit of the technology that we've been working on, as well as some of the kind of ways in which we've engaged with JGI in particular, and how this has really amplified the type of research that we've done within our lab. And as Ian has mentioned, mostly with a focus on guide RNA library generation and development and its applications. But first, obviously, we have motivating factors and different approaches that we take. So within our laboratory, we're very interested in the application of metabolic engineering, and it's really opened the possibility to make a lot of the fuels and chemicals that we want in a very sustainable, renewable type of manner, for everything from more sophisticated polymer precursors, commodity specialty chemicals, various different types of fuels, various other, not necessarily DOE relevant, but pharmaceutical and nutraceutical molecules. But at the same time, it's also opened up the wide variety of different organisms that we can modify and engineer and want to explore. However, at the same time, this pathway optimization, how we actually go in rewiring metabolism, getting from our starting carbon source of interest down to our product of interest, is something that's relatively complex. It's a biooptimization problem that has to really deal with this coordinated interwoven network of metabolites and enzymes and regulatory factors, some of which are known, some of which are unknown. And really that's been the motivating factor of some of the work within our laboratory, trying to find ways in which to navigate through this type of complex network. So starting off, leading up to the science getting toward the JGI CSP project, we were developing technologies in a one-off type of fashion, really looking at trying to systematically test what happens when we change the amount of a gene dosing, essentially. So looking at this enzyme perturbation sensitivity. So we had found some technology in our lab that we can use CRISPR-Cas9-based systems and use that to both program, later multiplex, but initially singleplex, the activation and repression of genes of interest. So we can actually go through and query one at a time the various different impacts that overexpressing and repressing an enzyme would have. So to do that, we actually found that within Saccharomyces, to start with, we can actually step or move the position of our Cas9 assembly to be able to give a large dynamic range. So essentially here, if we start with a repressor that's bound very close to the transcription start site, we can see the bottom here that we have very low expression overall. As we move that repressor further away from the start site, we can begin to slowly increase the amount, so decrease the amount of repression. And then if we were to swap this for an activator instead, and then step that closer and closer to that start codon, we can actually begin to activate its expression. So really this stepping is able to give us this graded effect from almost full repression to very high level activation. So we use this to think about this as turning a knob essentially on the overall gene expression for single genes of interest. So we can use this to ultimately program and multiplex. We can create libraries or cascades of various different types of targets that can then sit in the freezer. We can then pull these back out, always be able to query this in the same way. We have different degrees of actuation, whether it's positive or negative in terms of the activation or repression, and we can then assess a phenotypic response. So this is the lead-up science that we had going into this project, and really what we did was then wanted to expand this much more. How do we expand beyond just single one-at-a-time type of approaches? And this really required larger library scale synthesis, and this is where we started coupling together with JGI, especially the DNA synthesis group, to be able to enable this. And really this enabled us to get improvements both in terms of our multiplexing as well as our overall library coverage. We were no longer constrained to just a couple targets at a time. We really were able to expand beyond. So this led together to the formation of the first CSP project for DNA synthesis, where we brought together a team that was comprised of myself as the lead PI, Michael Jensen from DTU, and Huimin Zhao from University of Illinois, Urbana-Champaign, to work together to look at global sgRNA tiling across Saccharomyces. So a little bit of how we got involved in this and even learned about the process. So webinars like this didn't really fully exist back then, but we really first heard about the jgi synthesis grant during a presentation that Yasuo gave back in 2016. So this kind of was the sort of near the beginning of being able to do this DNA synthesis, but certainly the beginning of doing sgRNA libraries overall for jgi. So after hearing that and having this idea of being able to take this approach and trying to scale it up, we started discussing project ideas and scope for the overall development for Saccharomyces. That then led to us submitting a DNA synthesis grant similar to the one that's due in a couple weeks from now, back in July 2016. We then went through a series of contracting steps after we were selected for that, that completed by January 2017. And then it was a interactive back and forth manner. Really, we were able to work together with the JGI scientists along with planning meetings to discuss how we would want to develop this overall library. And then finally, after a couple of design iterations, we had our library shipped back to us in 2018 in January, and we're able to really implement that at the library scale. So this really designed the high throughput information. We were able to go from a stoichiometric model. We didn't have the sophisticated tools that Ian had talked about a couple minutes ago in terms of defining the guide RNAs design. So we had to go through and design those and ultimately created about 4,000 guide RNAs that were able to, in several different pools, target some of the major aspects of metabolism so we can actually tune those all up and down. Later, we used that in a similar fashion to what Ian had just talked about in terms of bringing these pools together, doing different enrichment steps, both for growth enrichment as well as for production, and then link that together with deep sequencing to be able to find the enrichments that we would have both in terms of overexpressions as well as repressions. This really amplified the research from what we would have been able to do. It enhanced the research that we were doing as part of the Air Force grant that we had for these overall individual guide RNAs, and recently resulted in a joint paper that was accepted just last week at PNAS. So really leveraging the JGI capacity with our overall scientific goals in mind. So we also had a very similar parallel project also in 2016 with Uroea lipolytica. This one was led by Ian Wilden at UC Riverside and collaboration also with Mark Berliner at Clemson University. And it was really a similar type of process where we had the project ideation, discussion with JGI staff, submission of the proposal, and then back and forth for the library generation and delivery. This was a slightly different, non-conventional host, but had several of its own challenges associated with that, but really demonstrated that sgRNA libraries were possible to be synthesized at JGI and were highly effective for functional genomics type of screens. So finally, as we move on to the future, we have continued our collaborations with JGI. It's a really great partnership and really great scientists to work with. And this is in a community science project aimed at being able to expand these technologies further, led initially by Ian Wilden, along with myself and Carrie Eckert from Colorado Boulder. And there it's looking at trying to expand these tools, really creating user tools for a variety of non-conventional yeasts that have importance for biofuel applications, and really comprises validating sgRNA libraries, rapidly synthesizing them, and then analyzing genome-wide functional genomics so that then that can feed into the bioinformatics pipeline of K-base and other DOE resources. So with that, I'd like to just really thank the people in the lab who have worked on this project, and especially for JGI for allowing us to be a partner of collaboration and amplify the type of research that can go on in our laboratory. So we have taken these projects in many directions that would not have been possible without the support from JGI. So with that, I think probably turn it over to see if there are any questions for a question and answer session.
Thanks, Al. We haven't had any other questions come in just yet, but we were curious about if there are actually any of the other platforms or capabilities that we offer that you could imagine combining or using in future studies or existing studies.
Sure. I think there are quite a bit, right? So this is, while it's a tool, at least within the projects that I just talked about, while it is a tool for functional genomics, we can also do quite a bit of characterization as well. So the opportunities and expertise that people have at Slack and other types of resources through JGI would enable us to do more than just a functional genomics stream, but functional metabolomics and functional proteomics, and really increase the depth of understanding that we have about these rewired metabolism in many of these cases, and also our understanding of these organisms in general, as we hope to harness them both as fuel producing organisms, but also understanding their natural niche within the environment.
That's great, thank you. We're going to move on then now to the next technology, but I just wanted to remind folks, if you have any questions for Hal or any of the JGI scientists, we'll have another chance to address those questions. So please feel free to just type them in the chat bar and we'll get to them. Okay, let's move on to Yasuo to give a background on the next bit of technology.
All right, let me share my screen. Okay, my name is Yasuo Yoshikuni, head of DNA synthesis science program at the JGI. The second session of this webinar would like to focus on genomes to structure and function, which is one of our strategic focus area in functional genomics call. People are usually interested in large-scale characterization of enzymes and other proteins, including binding proteins, transporters, sensor proteins, people who are interested in this genome to structure function category proposal. Typically, they're interested in discovery of enzymes and pathways with the new functions, discovery of proteins with desired function for synthetic biology, metabolic engineering, study of sequence, structure, function relationships. This is typically how we work with users. Users submit the proposal. When it's approved, they send large number of sequence to us. We then design the construct, build the construct. We deliver the construct to our users. Users then transform those constructs into E. coli, for example, then express them, purify, and functionally characterize them. In the functional genomics goal, we added the new capability to this pipeline. The first one, you can actually now send back your samples to JGI to do high resolution mass spec to characterize enzymes and pathways that you're interested in. Also, you could request the JGI to do sequence data mining and analysis based on our public databases, including IMG, Microcosm, and Phytosome. Also, we are currently working at a pilot program. We are trying to develop a pipeline working with the Slack, ALS, Advanced Photon Source, NSLS2 at the Brookhaven National Lab, trying to create the seamless pipeline to study all the way from genomics to function and structures. With that, I would like to introduce second speaker of the session webinar, Philipp Zerbe. He's a professor at the Department of Biology, University of California, Davis. He joined the UC Davis 2014. His research focuses are discovery and engineering of specialized terpene metabolisms in bioenergy, food, medicinal plants. The goal for his group are to develop tools of production of terpenoids with a human benefit. Prior to joining the UC Davis, Philipp did his PhD at the Ruhr University, Bochum, Germany, with emphasis on structure-function relationship in jasmonic biosynthesis. He then did postdoc at the University of British Columbia, where he investigated terpene metabolism pathways for stress tolerance in food and forest trees. I met Philipp at the first time in the Terpenoid conference in 2015. I was quite impressed by his research talk and discussed how JGI can potentially help his research project. He quickly assembled a team of scientists, as you can see here. and became the first recipient of our consortium type programs, where we can offer 3 times more than Moore's DNA synthesis capacity than usual route. We will hear more about his project and experience with working the JGI. Just a little bit more note, in recognition of his research contributions, Dr. Zerbe received the 2015 Arthur Nash Young Investigator Award, 2016 Hellman Young Investigator Fellow, and the 2018 Phytochemical Society of North America Young Investigator Award. And most importantly to us, he is the recipient of DNA Audi Career Award in 2018, which I believe that JGI Synthesis Award tremendously helped. We are very excited to hear how he has been working, successfully developing his science programs in Korea through the collaboration with the JGI. With that, Philipp, please take it over.
Just share my screen. Thank you so much, Yasuo, for the kind introduction and for having me join today and share some of the work that we've been doing collaboratively with the JGI over the past few years. As Yasuo said, I moved to UC Davis in 2014, and sort of the work with the JGI was one of the first things we actually started doing when starting the lab, and it has been, especially from an early career perspective, been absolutely instrumental in a lot of the work that we're doing now. And so just to give you a brief overview, my lab largely focuses on understanding the biosynthesis, function, and engineering of plant specialized metabolism, and in particular terpenoid metabolism, across a range of bioenergy, food, and medicinal plants. We have some projects in the pathway discovery and engineering for therapeutic plant natural products. Some more work going on in understanding the genetics and biochemistry of aroma metabolism to support crop breeding for flavor and aroma traits. But really the major focus of my lab is to understand the roles of terpenoids in mediating plant environmental interactions in bioenergy and food crops. And so this topic is really what we have built upon our collaborations with the JGI. And so we were fortunate to have 2 projects awarded by the JGI over the past 5 years. And much like what Hal described and what Yasuo just mentioned, is they really were built upon discussions with Yasuo and other people at the JGI to develop a strategy for a project that then eventually led to a proposal, and that eventually was supported to start the work. And so this first project that I wanted to talk about and show you was a consortium type DNA synthesis award that now is the Functional Genomics program, where we brought together a team of 6 labs, actually, Dorothea Toll at Virginia Tech, Björn Hamberger at Michigan State, Jörg Bohlmann at the University of British Columbia in Canada, and then 2 researchers, Eric Schmelz and Alyssa Hofacker at the UC California, San Diego. And so we brought these people together to really do a broad approach of trying to understand how different bioenergy relevant plants can produce species-specific blends of terpenoids. And I'll speak about the structure program a little later. So the background of our interest here in this grant was to better understand terpenoid metabolism across a broader range of plants. And that was important to us because, especially in monocot crops, and it's been well established, for example, for maize and rice, terpenoids play a key role as components of plant defense, both above and below ground, as well as many other plant environment interactions, such as allelopathic interactions, but also, as we now see, more and more evidence for potential cooperative plant microbiome interactions. So we're very interested to understand how these different species can develop species-specific blends of terpenoids and how they deploy them for the various different interactions with the environment to ultimately use this knowledge, for example, for crop optimization. And so this was a general goal, really understanding the biosynthesis and the diversity of species-specific terpenoid networks across a number of different species. And so to do so, we specifically focused in on two critical enzyme classes that define the diversity of terpenoids. These are the terpene synthetases, or TPS here, that catalyze the initial critical cyclization reactions. And then downstream from there, the large class of cytochrome P450 enzymes that facilitate a number of functional modifications of these natural products to eventually develop the range of different bioactivities. And just to highlight how this works, these pathways are actually modular pathway networks where two different kinds of class 2 and class 1 terpene synthases can work in different combinations of pairwise reactions to generate a number of different natural product terpenoid scaffolds. And these then undergo a number of different functional decorations, most commonly different oxygenations by the P450s. So these are very modular combinatorial type pathways. And so this modular nature of these pathways, we actually exploited for our grant to try and develop a platform to more efficiently identify these biosynthetic pathways. So how we went about this and really collaboratively built the strategy together with Yasuo and other people at the JGI was that we started out with a gene discovery platform that we had developed in my lab that allows us to screen large-scale genome and transcriptome resources specifically for terpene synthases and P450 candidates. We combined this then both with metabolomics and transcriptomic studies to prioritize gene candidates through correlation analysis, looking for tissue specificity as well as different patterns of stress inducibility. And so this pool then of prioritized gene candidates we shared with the JGI, who helped us out then with the DNA synthesis of all these gene constructs. And so in the end, because this was a larger scale consortium grant, it allowed us to actually synthesize several hundred genes at this time. They were synthesized by the JGI, then subcloned into the required expression vectors. And so what we received back very efficiently was the ready-to-use expression clones that we then could use for a system that I will show you in the next slide, where we can use combinatorial functional assays to understand these different terpenoid pathways. And as I mentioned, this was a consortium type grant. So we had 6 labs involved that focused on different plant systems. My lab focused on switchgrass, maize, as well as millet. Then Dorothea Toll's lab looked into switchgrass. The group at UC San Diego focused on maize. The MSU group focused on paper search. And then the Canada group from British Columbia focused on forest trees. But despite this different focus, we had a system in place for this large grant to actually openly share all the constructs that were provided by the JGI between these groups. And not just the constructs, but we also regularly exchanged functional logic that was developed between the groups so that we could quickly work with the enzyme functions that we were identifying to accelerate the pathway discovery across all these different groups. So it was a very efficient way for us, by openly sharing the constructs and the knowledge in this program, to try and find and develop a platform to identify complex metabolic pathways. And so the way this combinatorial functional assay system works is that we received now the gene constructs from the JGI. So these are multi-gene plasmids. Just as an example here, we could combine different variations of class 2, class 1 terpene synthases, as well as P450s. And these were then co-expressed in either E. coli, in yeast, or in Nicotiana benthamiana in tobacco. The reason for us using all these 3 different expression systems is to overcome any limitations in protein expression, which is very common, especially for cytochrome P450s. So this was very helpful to actually simultaneously run different expression platforms. And again, the capacity for us to synthesize several hundred genes made it also possible to actually synthesize them in a codon-optimized form for each of these different expression systems, which would have taken us a long time to actually do in-house. So it was fundamental to have the JGI support here. And then in order to actually analyze the enzyme products, we directly extract from the leaf material or from the cultures, and we're using mass spectrometry and or NMR to verify the enzyme products and understand different pathway branches and pathway combinations. If you're interested in this technology, in this platform we developed, we actually have a video protocol that's published in Jove that explains how we actually go about this expression system. This can be used for function analysis of enzymes, as I described. It's also fairly easy to upscale to larger culture volumes or plant biomass volumes to produce and purify these compounds for other types of assays. So we applied this approach now to all the synthesized contracts. And just to highlight one example here in maize, where this actually allowed us to really efficiently within 3 years to identify the vast majority of the diterpene syntases and cytochrome P450s that are relevant for the production of the species-specific diterpenoid blends in maize. So here we have in blue and yellow these combinations of class 2 and class 1 diterpene syntase pairs that produce all these different backbones. And then we identified a number of cytochrome P450s here that are multifunctional. As you can see, they function actually in several of these pathway branches to produce these different, now functionally modified and bioactive compounds. And what's really exciting about this approach was that we not only could identify the pathways to known compounds, such as the gibberellins as plant hormones, or the correlexins here as very potent antimicrobial compounds in maize. This approach actually also allowed us to identify new types of compounds that hadn't been discovered yet in maize, including this group here of dolabalexins. And so this was really exciting to see that this combinatorial DNA synthesis and combinatorial functional assay approach allowed us to identify potentially new natural products that exist in monocot crops. And so it was very interesting to us to now see and verify that these enzyme-based studies hold true also in the plant, and that these compounds that we identified are relevant in the plant. So we started to combine our DNA synthesis and functional study with larger scale metabolomics that were partly done in-house and partly in collaboration with the JGI metabolomics platform, a project that we designed with Trent Norton, who is running the JGI metabolomics platform. And so this actually then allowed us to show that these dolabalexins indeed are accumulating in field-grown roots of different maize varieties shown here. So they're indeed relevant in planter. And we could then further use our combinatorial assay system. We upscaled it to 20-liter E. coli cultures that allowed us to purify milligram amounts of these dolabalexin compounds. And we could show using these compounds that they are very potent in inhibiting pathogen growth, as shown here for a major maize pathogen, Fusarium verticillitis. And you can see here in this graph, this is just fungal growth over time. And even at fairly low concentration below what we find in maize root tissue, they already significantly inhibit fungal growth. So I'm just trying to highlight here that combining now the data we could generate based on our DNA synthesis award with both metabolomics and bioassay approaches really allowed us to understand the function of these newly identified compounds. Just to quickly move on to another example here, we did the same approach also in switchgrass, which at the time, terpenoid metabolism in switchgrass had not been studied at all. And in switchgrass, similar, we could use our combinatorial assay approach, and we had identified an even larger modular network of terpenoid biosynthesis. The gene family is 3 times as large as in maize. It's producing a large diversity here of different compounds that are produced through these different modular type pathway branches. And it is also producing a number of compounds that had not been seen in any other plant system. So an immensely diverse diterpene biosynthetic network that we could establish using DNA synthesis and combinatorial assays. And similarly, we went then into the plant. Again, we worked together with Trent Norton and the metabolomics platform, and we could actually show that several of these metabolites that we had identified accumulate in response to drought stress in switchgrass. So unlike maize, there seems to be a role more in abiotic stress responses. And so this data now we are using for a current collaboration with Trent Northen's group, where we use EcoFab chambers, so small controlled environment chambers that allow the plant to grow on these small 3D printed plates here, and allows us to feed in different solutions and extract solutions in real time while the plant is growing. And our first data that really are hot off the press from last week, we could actually see that some of these drought-inducible terpenoids are accumulating in switchgrass, and they're actually extruded into the rhizosphere here. So very exciting to now build into a new collaboration and really try to understand the physiological relevance and function of these compounds. So in summary, having started this project with the JGI early on when I started the lab, it has been absolutely foundational for much of the work we do now. Through this grant, we could identify the gene families for terpenoid metabolism in millet, in maize, as well as in switchgrass. We could underscore that especially the monocot crops shown here are very rich in terpenoids with much variation in the gene family size and in the diversity of species-specific terpenoids that are being produced. So it gives us an excellent basis to really try and understand their species-specific functions in crops as a resource for crop optimization. And as Yasuo mentioned, the data that we generated through this project have been really instrumental to then secure larger grants, including DOE and NSF funding that specifically target the understanding of terpenoid functions in crop environment interactions. And just very briefly to highlight another program that we're collaborating on with the JGI is the JG structure program as a pilot project, where we're working with Paul Adams at UC Berkeley and try to understand the structure function that is underlying the functional differences in terpene synthesis. And just to briefly highlight what we're doing here, so we're building on the enzyme functions that we had developed in our DNA synthesis grant. The JGI has helped again with gene synthesis for optimized constructs for protein production and purification. This is then done in Paul Adams' group that are doing the expression, crystallization, and X-ray structure solution of select terpene synthesis. And so the structures then go back to my lab, where we're really trying to understand the underlying reaction mechanisms that lead to different terpene synthase functions to eventually be able to actually engineer these functions. And just to highlight sort of the first structure that now has come out of this collaboration from a plant species that producing a very unusual diterpenoid precursor. And we're currently working with gene synthesis and structure-guided mutagenesis to actually understand which active site determinants define product specificity, and to understand both how these functions have evolved and how we can use this knowledge to engineer potentially novel diterpenoid scaffolds. So I hope this is helpful to sort of understand what all is possible with the different JGI platforms and the collaborations. I really want to thank Yasuo and everyone at the JGI for fantastic collaborative projects over the last 5 years. And of course, thank my team for doing all the hard work on it. And I'll be more than happy to address any questions.
Great, thank you for that talk. We do have a chance for some questions. I have a question that just came in saying, this is for Philipp. Can you share the catalog number for the 3D printed plant culture plates?
I don't have a catalog number. So these ecofabs have been developed in Trent's Northern labs at the JGI. And so what we've done in collaboration with him is that we are using the ecofabs that he has 3D printed. So I wanted to highlight it here because I think that's a development that's fairly recent. So it'd probably be good to reach out to Trent Northern's lab for the details. I'm not aware of a catalog number.
Yeah, it sounds like those ecofabs are custom jobs. So if you're interested in them, Trent Northern is the guy. He's a scientist affiliated with JGI and Lawrence Berkeley National Lab, and please contact him with questions about that. I have a question also, Philip. This is a little bit more general, but you mentioned a number of different types of proposals, but including one that was a consortium. And I was wondering if you could explain a little bit more from your perspective the difference between a consortium versus our more standard proposals that might have, it might have one or two PIs on it.
Yeah, I mean, Yasuo, please feel free to chime in as you know more of the details. But in general, the consortium type approaches are for bringing together larger teams of different labs to address potentially broader questions. And so in doing so, it also allows for larger scale of different of the JGI resources. In our case, it was a larger number of genes that we could synthesize. I think two really beneficial points for the consortium type grants was that it really facilitated networking across a larger number of labs. It greatly benefited student training across these labs, and it allowed us to address our question about diversity of terpenoid metabolism across a larger scale of plant systems that we would not have been able to address with a single lab proposal. And so this was something that evolved from discussions with Yasuo, from which sort of the scope of the proposal really grew with this opportunity to build a consortium.
Great, thank you. I was also curious if you could describe just a little bit more about, again, we're different from other agencies where you don't get money, you get resources, and we talk about access to capability and expertise. In your case, with DNA synthesis, could you explain, like, what physically are you actually receiving from us? Can you just describe that a little bit more? Or is it data? Is it a plate of material? What is it?
In our case, what we received were the functional constructs as cryostocks. So essentially, we developed the strategy for gene synthesis together with Yasuo and the JGI. And what we received were the functional expression constructs. So basically, we could accelerate all the gene synthesis, subcloning steps to actually get to a construct that we can use for expression. That was done by the JGI. So our lab could readily focus on the functional assays. So we actually received physical plates of cryostocks of the ready-made constructs.
Great, thanks. I have a couple more questions these are more general questions actually for Ian or Yasuo, if I could pass some of these questions over to you guys. Ian, actually, I guess a similar question though with the guide RNA technology. What is it that people physically are receiving from you? What is the product they're providing?
If you submit a successful proposal for a guide RNA library, what you will receive in a FedEx package is the sequenced Q-seed mixed culture containing of E. coli containing all of the constructs representing the full library.
Okay, great, thank you. I have a couple more questions. They're actually more general related to this, just the call for proposals. So David, if you wouldn't mind putting up that slide that summarizes this call for proposal, that would help. But before we get to that too, I wanted to I had a note here that I want to make sure that the attendees see in the chat, which was related to the question of the difference between consortium versus a standard proposal, and that is that the consortium proposals, they are permitted to request up to 1.5 megabases of synthesis compared to 0.5 megabases for a single lab. So you're actually able to scale up the request size to deal with these larger projects. Okay, so a couple more questions again about just sort of the general call for proposals. The slide is up again. This next call is due on July 31st, but remember that, and we have this call for functional genomics twice a year. And then it's also possible to access these capabilities in some of these other calls, like I said, the annual CSP or the FICUS proposal, which was mentioned in the chat bar earlier. I have a question for Yasuo just about the general process. I was wondering if you could explain a little bit about the review process that proposals go through, and also mention any rough ideas about the success rates.
Sure. So when the proposal was submitted to the JGI, so JGI scientists will go over the whole proposal for the technical feasibility first. And also, like, users are recommended to contact one of us to see what you are thinking about is very different from what we offer. You can always welcome to discuss and ask and see if we can do it, help build some of the construct that you'd be interested in ahead of time. After the technical review, we'll be sending those review to peer review. Those are very similar to regular grant application approval process. And then once the review panel recommends proposals for acceptance, we'll then send it off to internal review process, which we look into biosecurity, biosafety, environmental aspects of the proposals. I think nowadays, especially in synthetic biology, those are very critical things to consider. And then finally, we'll get the JGI selectors approval and also DOE project managers approval. And we'll contact back to you for actual contract, and work will begin.
Okay, thank you, Yasuo. Appreciate that that background. Well, we're in the home stretch here, and I'm looking to see if there are any other questions that have come through the chat that we haven't been able to address yet. It doesn't look like it, so I'm going to say thanks to our guest speakers, Hal and Philip. We really appreciate you telling us about your work and how JGI was able to collaborate with you on it. Thanks for those of you online who participated in today's events. If you have, we're going to post some answers to the questions that we received. If you have any others, please feel free to reach out to contact Ian, Yasuo, anyone on here. There's also an email at the bottom of the slide right now to the JGI Communications Group. So if you have follow-up questions that you would like us to address, please send those over. And stay tuned. We've recorded this, so stay tuned for info on how to access this archive if you're interested in going back or sharing with anyone who might want to see it. I wanted to point out that shortly or later today, we hope to send out a survey about this. And so a question about what you learned today and what you might hope to learn in the future, and a number of different questions. And so I really hope you can take a look at that survey and give us some feedback, and because we hope to do more of these things and to really, improve how we can communicate what it is we have at the JGI. So one last time, the deadline, July 31st. I know it's coming quick, so if you're interested, go for it. Finally, I guess I think we lost that last slide, but I was just going to remind folks that if you want to stay in touch with the JGI, the latest from the JGI, you can follow us on any and all of the various media platforms of Facebook and Twitter, LinkedIn. And we also, I wanted to point out, we have 2 podcasts that we've been producing. One that's focused on secondary metabolism and bioproducts, and another just on sort of broader category of JGI projects. I actually enjoy these podcasts myself. I put them on when I go walk the dog in the morning, and check them out. They're pretty cool. Okay, well, that does it for today. I hope that we were able to provide some info, and good luck with crafting your proposals. Reach out to us if you have any questions. And that's it for us today. Thanks a lot.